Welcome student. In molecular basis of inheritance, today we discuss that is the topic of t-RNA, that is the transfer RNA. Transfer RNA, which is also called the adapter molecule. And why it is called the adapter molecule? Because it adapts the correct amino acid or it adapts the specific amino acid. That the question which is who read the genetic code? We know very well that the genetic code, that is the codon, and who read that codon, who read the genetic code and the result in the form of the arrange, which is the in the arrangement of the link of amino acid, and the answer of that question which is the t-RNA, which read the genetic code, t-RNA which read the codon and they give or they arrange the specific amino acid, that is the transfer RNA, which is the smallest RNA, and smallest RNA, that is the 10 to 15% of total RNA in the cell. That is the total percent. What is total percent? That is the 10 raised to 15. That's why it is the smallest RNA. And it's synthesized in the nucleus. ह्याला कोण तयार करते किंवा हे कुठे तयार होते? It's synthesized in the nucleus by DNA. The DNA which made the RNA, and in the nucleus it is also called the sRNA. It is also called the sRNA because sRNA, that is the soluble RNA, because it is soluble in 1 molar NaCl sodium chloride solution. That's why it is called the soluble RNA. That's why it is called the sRNA. tRNA, which is the adapter molecule, tRNA, which is the sRNA, it is a single stranded RNA. जसं की DNA हे डबल स्ट्रँडेड आहे, but RNA which is a single stranded RNA, and with 75 to 90 nucleotides are present in that of single RNA. That is the single stranded RNA, and that is the prime end, that is the fifth to third prime end of the sequence of that RNA. And because of the folding structure, it look like a double stranded दिसतं, पण कधी ज्यावेळेस त्याच्यामध्ये फोल्डिंग तयार होते, because of the folding it look like a double stranded, because of the folding it looks like a clover leaf model, because of the folding structure. This structure it's look like this type. Because of the folding, this structure it looks like this type. Because of folding, these are the loops and these are the hydrogen bonds which are look like a double stranded structure. This discover, that is Hogland and Stephenson, and it acts as a carrier of amino acid. काय काम आहे ह्याचं? It acts as a carrier of amino acid during photosynthesis, and according to Francis there is a mechanism. Francis Crickच्या मते काय आहे? According to the Francis Crick, there is a mechanism. That mechanism which is, there is the mechanism that there is a codon that read the specific codon. There is a mechanism that read the codon and arrange the amino acid, and that mechanism to the molecule, that molecule or that component which is the transfer RNA. Now we see the structure of the tRNA. It is transfer RNA. It is a single stranded, but because of folding it look like a double stranded RNA, and it look like a structure that is the clover leaf structure. It is the clover bonds. Hydrogen bonds. Because of folding, these hydrogen bonds are formed, but the unusual nucleotides or the abnormal nucleotides or abnormal nitrogen bases here, there is not hydrogen bonding are present at some, that is some distance here. That's why there is a loop like structure are formed. इथं का हायड्रोजन बॉन्डिंग नाही आहे त्याच्यामुळे काय झालेलं आहे, the loop like structure are formed here because of abnormal sequence of nitrogen bases. Here the nitrogen bases sequence are the complementary nitrogen bases sequence, but here abnormal bases here. That's why here not forming a hydrogen bond or not look like this type of structure, here look like this type of structure here, and that structure which is called the loop, and each and every loop here one specific name here, that is this loop is the DHU loop. First of all there are RNA t-RNA which is the single stranded and one end is fifth end and second end is third end, which is always that is the direction fifth to third. Here that is this is the fifth direction, and here the end of that direction that is the third end, and fifth end that is the nitrogen base is present, that is the guanine, which is the guanine nitrogen base is present here, that is the fifth prime end. And this loop that is the called first loop which is the DHU loop, this loop which is the dihydrouridine loop, dihydrouridine loop, this loop DHU dihydrouridine loop, and it helps to recognition enzyme which carry the amino acid, and that enzyme is amino acid t-RNA synthetase enzyme. This loop which helps to recognition that is recognition of which carry the amino acid, that enzyme which is called the amino acid t-RNA synthetase enzyme. This first loop. Then the second loop which is the anticodon loop. Why it is anticodon loop? Because of it read the mRNA and mRNA codon. And why it is called the anticodon? Because it is the complementary sequence or complementary bases here that understand that is the tRNA, which is most important function, that is read the codon